The next section in our chapter is on chromosomes and DNA replication. We went over two main types of cells, prokaryotes or eukaryotes. Prokaryotes have no nucleus. They are more simple. They have one circular DNA strand. It's very easy to copy. With us, it becomes a little more complicated. We covered that in mitosis. So with that, your DNA replication is part of the S phase, if you remember. So when we look at eukaryotes, they're more complicated. DNA is inside of a nucleus. It's kept safe there. When you need information from DNA, they actually make little copies and send it out. If you look, we have different chromosome numbers depending on an individual. We looked at human karyotypes, so you should remember that humans have 46 chromosomes, fruit flies have 8, and giant sequoia trees have 22. If you look at the length of DNA, it depends on the organism. A prokaryote, which is a bacteria called E. coli, has one chromosome that is 4,639,221 base pairs long. This is only about 1.6 millimeters long, so it's very small. Human chromosomes are bigger, a lot more complicated. Our eukaryotic chromosomes also contain both DNA and protein, and they're tightly packed together to form what we call chromatin, another word you have heard of. When you look at that composition, the DNA and the proteins that are involved are called histones. So here's a picture. You can see that our lab that we did talked about DNA in this double helix image. You have two base pairs, maybe A and T and C and G, and they're always paired together. Once those are coiled up around these proteins that are called histones, that coils up even more, becomes what they call nucleosomes, coils, eventually getting to the chromosome. Remember, we have 46 of these chromosomes, so that's a lot of DNA. When we make copies of that right before we split cells in mitosis, it's called DNA replication, the scientific term for the copying of DNA. You might also hear the word DNA synthesis, because synthesis means to make. So in order to replicate DNA, you have to do a couple things. First, the DNA molecule has to be separated or unzipped into two strands. And then those two strands, or the sides of the ladder, they're each used to make, as a template to make the other side. And you wind up having two strands, half of which are part old and part new copy. Here's a picture of it. You see this is in the middle of a strand. It's been unzipped, and you have the this blue here is the old strand, and the red that's coming in is the new strand. You can see two bluish green balls in the back, and that says DNA polymerase. That is an enzyme that's going to work and actually do the job. So that main enzyme that's involved is called DNA polymerase. This adds on the new nucleotides that fit into that sequence. And then it proofreads to make sure that there haven't been mistakes made. Once all of that is done correctly, the cell can then go on to mitosis and split to make two new cells. So if we remember, say you've eaten your lunch and you need to make some digestive enzymes, your body will need to go into your DNA and get the instructions to make those. So this would be the first step in creating anything in your body. So that is DNA replication.